sir.
So let us start.
Good morning, sir.
Good morning, sir.
Good morning. So today we shall be dealing with a topic, molecular phylogeny and phylogenetic analysis. Today I shall only take on the theoretical part, and then tomorrow at the same time I'll be taking on the practicals. If the time permits, today itself I shall start the practicals and I'll show you how to download the required sequences if there is time towards the end. But if not, then I'll limit myself towards the theoretical concept only today. So what is phylogenetics? So phylogenetics is a branch of biology that deals with establishing evolutionary relationship among organisms. So all species of Earth, according to biological evolution, have emerged due to certain evolutionary changes in the genotype and ultimately resulting in development of different strains and species. So by going back into the evolutionary relationship among the genotype of the organisms, it is now possible to find the ancestry of the species, and based on the ancestral origin, we can establish relationship among them. And that particular branch of science that deals with the establishing the evolutionary relationship among organisms is known as phylogenetics. And with the advent of the complete genome sequencing technology, now we have got the complete genome of a number of representatives of a species in the databases. And people have started thinking, establishing relationship among species based on the whole genome sequences rather than on the basis of comparing the sequences of certain conserved genes. And therefore, we are quickly moving towards phylogenomics from phylogenetics. So today we shall discuss very briefly about what are the principles and methods of phylogenetic analysis, and on the basis of the analysis, how we can draw a phylogenetic tree. So as I say, that the phylogenetic relationship among species can be established based on the genetic sequences represented by DNA and RNA. But of course, we can also take into account the protein sequences for this purpose. So before the advancement in molecular biology and sequencing technology, people also attempted to classify organisms based on empirical criteria, based on morphology, biochemical characteristics, and other empirical phenotypic characteristics. But when there was an advancement in molecular biology and sequencing technology, now we rely more on the molecules or the biomolecules like DNA, RNA, or protein sequences rather than the empirical methods of phylogenetic classification. So DNA of the organisms in lines of descent actually accumulates mutation over evolutionary time. So mutation is a continuous process which occurs both spontaneously as well as under the influence of certain mutagenic factors in the environment. And this process leads to changes in genotype over a period of time. And due to accumulation of the result of mutation, there can be significant impact on the phenotype, resulting in observable changes in the physical, biochemical, and other characteristics in the phenotype. And a new strain or species may emerge due to this evolutionary process. As the nucleic acids and proteins evolve, one can therefore very easily build inferences based on the molecular data regarding the genetic relationship or rather evolutionary relationship among the species or strains, and that is called phylogenetic analysis. Now, as I said that today we give more emphasis on using molecular data rather than the empirical data for phylogenetic analysis, because use of molecular data for this purpose has got some specific advantages. The molecular characters are discrete characters. As we know, that if we take into account the nucleic acids like DNA or RNA, say DNA sequence is composed of 4 variable characters or nucleotides, A, B, G, and C. Similarly, RNA is composed of A, U, G, and C. So these are discrete characters, and therefore they are unambiguous, and there is no chance of confusion. They are totally different from each other. There is no confusion regarding the position of one nucleotide in a gene sequence or genotype sequence. So molecular data therefore gives us a clear picture of the genetic makeup of the organisms without having any ambiguity or without having any confusion. Another advantage of using molecular data is that it can be converted easily to numerical form, and therefore we can apply the principles of mathematics and statistics to analyze the data. Be it nucleic acid data or amino acid sequence data, both are convertible to numerical values, and therefore we can apply the principles and methods of mathematics and statistics to analyze them to get a measurable quantification. Protein sequence data has got further advantage on the nucleic acid data because of certain facts. Number one, the protein alignments are often more informative because we have got 20 amino acid variants compared to 4 nucleotide variants, and therefore the variation or the diversity that can be expressed by 20 different variants of amino acids are more clearly visible than that of the nucleotide variation. The amino acids also share certain biophysical properties, as we have discussed in the last 2 or 3 classes, that they are grouped into different families based on their biophysical characters. And if a particular amino acid of a particular group is replaced by mutation by another amino acid of the same group having the same biophysical property, there may not be any impact on the phenotype. That we already know. And therefore, if we consider the protein sequences for phylogenetic analysis, it can give us more appropriate information regarding the phylogenetic relationship among species, because that has got direct correlation with the phenotype. but many of the genotypic changes caused by mutation may be masked and they may not reflect on the phenotype that we know. This is because of the fact that the codons are degenerate. That means, as we know, that for the same amino acid, there may be more than one codon, except for a very few amino acids, rather two amino acids, methionine and tryptophan. Other amino acids have got more than one codons, and most of them are having more than two codons. Say, for example, proline has got 4 codons, leucine has got 4 codons, isoleucine has got 3 codons, thymine has got 4 codons, like that. So you see, suppose in a particular amino acid codon, suppose for proline, CCU is a codon. If in the third position, there is a mutation of the nucleotide and U is replaced by C, A, or Z, there is no impact on the protein sequence because all these 4 variants of the codon code for the same amino acid proline. So same is true in case of many other amino acids. So because there is a degeneration in the codon, therefore changes in the third position usually do not reflect on the change in amino acid. And that is why many of the events of mutation occurring in the genotype may not have direct reflection on the phenotype. And therefore, amino acid change takes place, or rather the protein sequence change takes place only after a very long evolutionary time compared to the mutational events. So therefore, if we consider protein sequences for establishing phylogenetic relationship, then we can get a chance of going back into the evolutionary history by a longer time duration. Another important advantage of using protein sequence is that even if you have got only the nucleotide sequence, you can also translate it into a protein sequence by use of bioinformatics tools. But the reverse is not possible. From a protein sequence, you cannot get back the DNA sequence which code for the protein sequence. It is because of the fact that there may be use of different codons for the same amino acid. So in fact, according to species, there can be a different preference for specific codons for an amino acid. And that particular study is called codon bias. So there can be bias among the species for using a specific codon for a specific amino acid. So that is another aspect of bioinformatics that can be studied, the variation in the codon bias among species. Now, although protein has got several advantages over the DNA sequences, but it has got also certain disadvantages. Proteins could be under evolutionary pressure. Proteins can be influenced by the environment and epigenetic factors. So, as we know, the transcription and translation process of a gene is influenced by several factors, including certain genetic and epigenetic factors. Similarly, the environment where the organism grows also influence tremendously on the types of proteins transcribed and translated at a given point of time under the influence of a given condition. Let me give an example. Say, for example, E. coli is a bacteria, Escherichia coli, which is known as a facultatively anaerobic bacteria. Means it can grow both in aerobic as well as anaerobic condition. A particular strain of Escherichia coli when we grow in aerobic condition, it will produce hundreds of different kinds of proteins which are required for its metabolic activity in the presence of oxygen in the aerobic condition. But all those proteins may not be synthesized by the same bacteria when it is grown in anaerobic condition, because it will require some other kinds of proteins and enzymes which will be essential for the survival and growth of the organism in absence of oxygen. So the protein content of the organism, although the genetic content is same, the protein content may vary according to the influence of the environment. Another disadvantage of using protein sequences for phylogenetic analysis is that, as we have already discussed, many of the mutational events that has taken place during evolution might have been missed when you use the protein data only for phylogenetic analysis, because many of the mutational events may not be reflected on the amino acid sequence. Therefore, today there is a tendency to use both genetic, that is your DNA or RNA sequences, as well as the protein sequences for phylogenetic analysis. But one condition is that the sequences that are used for phylogenetic analysis should represent some conserved sequences. In case of bacteria, the 16S ribosomal RNA sequences are known to be highly conserved within a species. That means when it undergoes mutational variation over a period of time, there is emergence of new species. So within a species, the variation in the 16S ribosomal RNA within a particular species of bacteria is very, very low. And therefore, this is usually considered as the most useful conserved sequence for identification of species of a bacteria. Similarly, in case of eukaryotes, the mitochondrial DNA sequences, as they originate from the mother, means they are maternally inherited. The entire mitochondria, in most cases, are derived from the ovum, and there is no contribution of the sperm. And therefore, the mitochondria usually carry the lineage of a species or a family. And therefore, the mitochondrial gene sequences are highly conserved in eukaryotes, and they are the sequences which are preferred for phylogenetic analysis. Among the proteins, say for example in bacteria, the ribosomal proteins, for that matter in eukaryotes also, that those ribosomal proteins can be used for phylogenetic classification. Now there are some known problems in using molecular data for sequence alignment and thereafter phylogenetic analysis. Actually, for any kind of phylogenetic analysis to be done, the first and foremost prerequisite is to go for a sequence alignment to find out the similarity or differences among the sequences. So if your phylogenetic analysis is to be correct, the first prerequisite is that you are to use the appropriate method of sequence alignment so that you can arrive at a inference regarding their similarity and dissimilarity based on the perfect sequence alignment. But whatever the procedure is used, in the usual procedure of phylogenetic analysis, normally most of the software tools account the variation of evolution rate at different sites of the same sequence to be occurring at the same rate. So, but normally it is not true. Within a genome sequence, the rate of mutation occurring in different sites or different loci or different genes will be variable. But this is not taken into account by most of the phylogenetic analytic software. This is one problem. Another problem is that there can be variation in the mutational rates depending upon the species or the taxa also. So there can be variation among species in the rate of mutation. And that is also usually not taken into consideration while doing phylogenetic analysis by conventional process. And this is another disadvantage or problem. Now there are various methods of doing phylogenetic analysis. Out of them, some are more commonly used or more popular. We shall discuss about some of them today. But when we have got a number of methods available for doing phylogenetic analysis, the question is, on what basis we shall select the appropriate method for phylogenetic analysis? Because selection of the method appropriately is the first prerequisite for a correct phylogenetic analysis, as I already referred to. So in this particular slide, I'm just showing how to decide about the best method for doing phylogenetic analysis for a particular set of sequences. So for any kind of phylogenetic analysis, we need to have a set of related sequences which are needed to be analyzed. First, we obtain the sets of sequences either from our own experimental data or from databases. We can download them from databases, and we can make a file containing all the sequences. Then we are to do the multiple sequence alignment by using some appropriate software. Either you can use Cluster W, Cluster X, Mega, Pop, and many other softwares can be used for doing the multiple sequence analysis. Again, doing the multiple sequence analysis in the correct way is also important, as I already mentioned. After doing the multiple sequence alignment, you are to analyze the alignment score and the result of alignment and ask this question: is there any strong sequence similarity? By looking into the aligned data, you are to answer to this query, whether there exists a large-scale similarity among the sequences. And if the answer is yes, then you are to select a method of phylogenetic analysis, what is known as maximum parsimony. Then if the answer to this query is no, that means you do not find large-scale similarity among the sequences, then the method of analysis will be distance matrix methods. And if the answer to this query is also no, that means if you have no large-scale similarity, second question will be: is there any clearly recognizable sequence similarity? Means, although there is no large-scale similarity, is there some segments where there is a similarity among the species? If it is clearly recognizable, then you have to follow distance matrix method. But if your answer to this question is also no, means there is no recognizable similarity at all, then you are to take on the third method, which is called maximum likelihood method. Whatever method that you use, finally, you are to analyze the data further for its validation, and ultimately, you are to decide about the correctness of the phylogenetic analysis. We shall discuss a little bit further on that, then it will be clear to you. So as I said, that three basic methods are more commonly used: maximum parsimony, distance matrix method or distance method, and maximum likelihood method. This has been already discussed in our previous lectures, you know now how to align the sequences and how to score the distance as well as similarity. For protein alignment, usually we take into account certain standard metrics. Sorry, there is a problem. Please wait a bit. I'm coming back. Yes. Is it available to now? Available to now? Have you seen the slide now?
Yes, sir.
Yes, sir.
Okay. So as I say, for protein sequence alignment, we usually use some standard matrices like PAM or BLOSSOM. So this is one example of the PAM 250 scoring matrix of Dayhoff. It was already explained to you. I'll not discuss it today. Now, distance matrix methods, as I referred to, there are more than one variations in the method. Most commonly, distance matrix methods are of two types: UPGMA, unweighted pair group method with arithmetic mean, and neighbor joining method. And of course, few more methods are available. Another method is Fitch-Margolius method. Out of these methods, UPGMA and neighbor joining methods are more commonly used. What is UPGMA? So this is one of the oldest and simplest methods of doing phylogenetic analysis. This method is based on statistics. It was first proposed in 1960. Now let me give you an example how this UPGMA method can be used for drawing phylogenetic tree. Suppose we have got a set of data of 4 populations, A, B, C, and D. So these populations may represent 4 variants of the same species or maybe 4 different species. Depending upon the situation, you have to take a set of sequences involved. So these are 4 different sets of data, A, B, C, and D. And we have already found by phylogenetic analysis what are the distance scores between the populations to each other. So A to B distance score was 4, A to C distance score was 9, A to D distance score was 20. Similarly, B to C 7, B to D 18, C to D 17. So based on these, the alignment score, how can you draw a phylogenetic tree by UPGMA method? First of all, our approach will be to identify the pair of data having the least distances. So out of these A, B, C, D sets of data, we find that A and B has got the least distance score, only 4. So we take into account this pair first. So distance between A and B is 4. So the average distance, it's a arithmetic mean of distance between A and B will be equal to total distance divided by 2. 4 by 2 is equal to 2. So therefore, we shall draw 2 lines here showing A and B. Both of them are connected by a horizontal line by first drawing 2 perpendicular lines of 2 units. This unit may be anything. It may be centimeter, millimeter, inches, whatever, or maybe in a graph square, whatever you can use. So we will first draw two perpendicular lines from A and B of the value of 2 unit, because their average distance is 2. Then we will join these perpendicular lines by a horizontal line. So that will depict the difference between A and B. Now, after knowing the difference between O and B, now we shall discuss the average difference or distance of A and B together from C or from D. So in that case, we shall consider A and B as a single unit. So distance of AB from C, A and B taken as a single unit from C, will be equal to distance of A and C plus distance of B and C divided by 2. So these values are already known to us. Distance of A and C is 9, distance of B and C is 7, and therefore it will be equal to 9 plus 7 divided by 2 is equal to 8. So from AB, the average distance of C is 8. So therefore, on that basis, we shall draw another line from C here, keeping the distance between A, B and C equally. We shall draw another perpendicular line of 4 unit. Okay, and then we will join this line to a perpendicular line drawn from the midpoint of the horizontal line between A and B, and we'll join them like this. And this picture will now depict the relationship between A, B, and C. Similarly, distance of A and B from D will be equal to distance of A to D plus distance of B to D divided by 2 is equal to 19. Okay, we will draw another, that means we will plot the values again, and then we will show this distance like this. And as I have already said, average distance of D from AB equal to 4, already we have drawn it. Then we shall go to the next. Average distance of D from ABC. That means this time we shall consider A, B, and C as single unit, as we did before for A and B. This time we'll consider A, B, and C as a single unit. So distance of ABC from D will be equal to distance of AB from D plus distance of C from D divided by 2. So it will be equivalent to, as we have already calculated, 19 plus 17 divided by 2 is equal to 18. So therefore, the average distance of D from ABC will be equal to 18 divided by 2 is equal to 9. So therefore, we shall draw another perpendicular line of 9 unit length, and then similarly we will join them with ABC. Now this is the final phylogenetic tree showing the variation between A, B, C, and D. So in this process, what we normally do, that means every time we calculate the distance between a pair of data, and among the different pairs, we find out the pair showing the least distance, and then take the average distance between them, and thereafter we group them subsequently by taking into account their subgrouping as 2 sets of data at a time, or 3 sets of data, or 4 sets of data, like that. We will proceed on, and every time we will be drawing the diagram by taking the average distance. So this is the way how the UPGMA method is used for drawing phylogenetic tree. So from this, we can easily say that A and B are more closely related than that of C and D. And C is more closely related to A and B than D. Another method of distance matrix method is neighbor joining method. This is not a statistical method, but this is a least square method. And this is done. So next we'll be discussing briefly about the neighbor joining method. So in the neighbor joining method, it is also based on the distance score as we discussed earlier. But here the procedure is little different from the previous one. It is not based on the arithmetic mean, but it is based on least square values. And here the neighbors are joined one after the other by this procedure. That means if A and B are more closely related, then the A and B will be joined together. Then the next close relative will be joined like this. Then the next relative. So this will go on. So because of the method involving joining neighbors one after the other, this method is called neighbor joining method. These are the two popular methods which are commonly used in phylogenetic analysis if you decide to take on the distance matrix method after sequence alignment. Now next we shall discuss about the validation of the phylogenetic analysis. After we perform the phylogenetic analysis using the appropriate method, another step is important, that is validation of the phylogenetic tree. There are more than one method of validation of the phylogenetic tree. One of the most commonly used method is called bootstrapping. So it will determine the reliability or acceptability of a phylogenetic tree. So in bootstrapping, actually the system will randomly select a variable number of the sequences from the set of sequences that you are taking for phylogenetic analysis again and again, and every time it will draw the diagram by following the same phylogenetic analysis procedure. Say for example, we are taking UPGMA method. By using the same UPGMA method over and over again, the phylogenetic analysis will be done by the system by picking different sets of values randomly from the total set of sequence. Say for example, we have taken 12 different sequences for a phylogenetic analysis. Sometimes the system will take 11 of them randomly, another set of 11 will be taken randomly second time, or maybe 10 at a time, 7 at a time, 6 at a time, and then every time by applying the same method, the system will try to develop the phylogenetic tree. This process will be repeated for about 500 to 1,000 times. And then, based on the results obtained, it will be determined out of 100 times, how many times a particular branch is appearing in the tree at the same place. So based on the percentage of occurrence of a particular branch in the phylogenetic tree, the validity will be determined. Normally we expect that the validity for each branch should be very high. If it approaches 100%, then it is always better, but never you will get 100%. When we get better values than 70%, it is considered to be a valid or reliable tree. Now another method of bootstrapping is Felsenstein's bootstrap test. I'll not discuss on this. This is again based on a statistical method of calculation of the validity. Now let me discuss about the terminology and the types of tree that are usually drawn. So basically there are two types of tree. One is called rooted tree and the other is called unrooted tree. Difference is that in the rooted tree, the ancestry is shown, the ancestor is shown. Suppose these two branches are linked to a root. A root stands for the ancestor. That means if these leaves represent two different species, both these species have evolved from the same ancestor, the same species. Okay, so each of the components linked to a branch is called a leaf, and they are joined together in a branch point. They may be linked to the root, or maybe they may be linked to another cluster of variable number of leaves. So a rooted tree is one in which the root is known or the ancestor is known. But in case of unrooted tree, the ancestor is not known. So this is one example. Unrooted tree. You see, there are no ancestor. The branches are linked together. All leaves in different species of components, but we don't have an ancestor. So this is called unrooted tree. Now there are some common terminology used in phylogenetic analysis. For example, node. A taxon unit. So is it under a species or string or any variant within a species? We differ from situation to situation. Sometimes when we want to draw phylogenetic conclusion within a species among different strains, each of the nodes can represent a strain. So node or leaf can also be used. Similarly, branch defines the relationship between the taxa in terms of descent and ancestry. A branch will define the relationship between the taxa and species or strain. Topology means the branching pattern. A branch length will represent number of changes that have occurred in a branch. So branch length is directly dependent upon the alignment scope. More scope, more the length of the branch. Root, as I already said, represents the ancestor. Distance scale represents number of branches between organisms or sequences. Then a clade. A clade means group of two or more taxa or DNA sequences or fragmentary sequences that include both their common ancestor and all their descendants. So for example, here in this phylogenetic tree, human mouse are forming they are making a clade, and their variation is minimum in this particular phylogenetic tree. Whereas the fly here represents a distant relative of human and mouse. The distance is opposite to the length of these branches. I already said the branch length will be the distance. And therefore, based on the type of information a tree will give us, they can be classified into different types. Already I told you, they can be classified as rooted or unrooted tree. And both rooted and unrooted tree can again be classified further as scaled and unscaled. So ultimately, we can have therefore trees which are rooted scaled or rooted unscaled, or unrooted scaled or unrooted unscaled. So a scale tree means we follow a primitive scale of measurement representing the phylogenetic distance shown by branches. So branch length is maintained in terms of the distance score that we calculate. This is one example where phylogenetic analysis was done based on mitochondrial DNA sequence of human, chimpanzee, gorilla, and orangutan. We know that these four species are known as humanoid apes. As you know that humans have evolved from certain humanoid apes, but in the development of genomics and sequencing technology, are not known whether human beings evolved from chimpanzee, gorilla, or orangutan. Of course, another humanoid ape is gibbon. 4 humanoid apes are available. They are all similar in their morphological, behavioral, and physiological properties that are human. But now, based on genetic analysis done, it has been established no doubt that human beings have evolved from chimpanzee. So chimpanzees are our closest relative. In diagram, to draw phylogenetic relationship among humanoid apes, we have another species, baboon, which is an ape, but not a humanoid ape. They are different from human being. We have kept this particular species in this analysis because in any phylogenetic analysis, they are advisable to keep an outlier or outgroup which is known to be not related to the other sequences. So that after the tree is drawn, we can see that the outlier or the outgroup is showing distant relation from the other sets of data. Here, baboon is not related to humanoid apps, so this is not considered a humanoid app. So after 23, therefore, it has shown maximum distance from others. So this is an outlier or outgroup. So used as one of the sets of data in the phylogenetic class, so that we can arrive at a conclusion regarding the correctness of the phylogenetic tree. If the outlier or outgroup shows high level of similarity with other members of this set of data, then we will know that there is something wrong in the phylogenetic analysis. For phylogenetic analysis, we require the sequence alignment done as a prerequisite, as I already said. Without doing sequence alignment, we cannot apply any phylogenetic analysis tool for drawing phylogenetic tree. So first of all, we require a set of related sequences, either sequences that are required to be analyzed. Then in alignment software, you can use either Clustal X or Clustal W, or you can use any sequence alignment software. You can use Muscle, the other software. Then you can use an effective phylogenetic analysis tool like Phylogeny, etc. So that is now a very popular tool, which is an open access tool, can be downloaded from the internet. So now, practicals, we shall be using Mega. So we'll be using Mega version 6.06. Of course, now the latest version of Mega again, the 6 is more user friendly. That's why we're using the 6. Then also for some softwares like Phylogeny, you need other software for viewing tree. After that, a tree view software is essential when you use Phylogeny, but it's not essential when you use Mega, because Mega itself is having a tree view software with it. So these are requirements for a phylogenetic analysis. When we will do it practically in the next class, then we will get to you. Now I will show you how to download devices doing this ready for phylogenetic analysis. So for that, we will find the simple slide. Use it as a file for uploading to any sequence alignment. Okay, we have gone to the NCBI database. Now, in our practical, we shall be using a mitochondrial gene sequence of different mammalian species downloaded from NCBI. So for that, we shall go to gene database. Okay? We shall go to gene database. So in the gene database, we shall search for COX-1 sequences. Cytochrome oxidase 1 sequence is described as COX-1. So without naming the species, we shall simply write COX-1 here, because we want to download COX-1 gene from different species, and we want to download the complete gene sequence. So, sorry, simply we have to write COX-1. COX-1. Wait a bit, still loading. So COX gene we shall try to download. So we have written here COX-1, then stops. Find it. We have selected gene database, and then we have written COX-1 here, so that we can get the complete gene sequences of the cytochrome oxidase 1. So as default, we use a list of different. So there are 61 pages. There are 20 items, and then 1,271 sequences in available in the database. That means you know on that the sequence of 12,271 RNAs are already available in the NCBI databases. So out of them, we shall select some of the some gene sequences. I think you will why because I think that bioinformatics need to have some consequences which are normally conserved in space. So COX1 or cytosine is a gene sequence, and this is highly important within a space. Now the first one is space is not space. We will take one. We do not want the starting function, but one the sequence. So that is a big. First is zebu cattle or so the details of the Bosch Torus sequence is here, but we need to find out the FASTA sequence, the entire gene sequence of COX1 gene. Here there are descriptions. I'll not go into details about that, but let me tell you briefly some of the features shown here. You see it is given gene symbol COX1, gene description, cytochrome C oxidase, subunit 1, gene type, protein coding gene, then organism is Bosch Torus, then lineage means the taxonomic position, it is writing the all taxonomic details, and then here it has given us the gene map. So this is the position of the COX1 gene in the entire genetic map of the mitochondria of Bosch Torus. So if we want to download the sequence, then we'll have to go down to the bottom. And here France Lawson relative number of the particular gene. So called FASTA. If you click on this FASTA, you will get the gene sequence. The gene sequence. So we can get mitochondrial genome. But this does not depend on the genome. We have no on these. How we enter mitochondrial genome? This is data or part of mitochondrial genome where the phosphate is located. So this stresses from the nucleic number 87 up to 7231 of the entire mitochondrial genome. So this is we will set this. We have selected it, then click it. We have clicked boot peptide. Whatever is a boot, you will click it. I will this option. This option. It is asking what is a complete record. Okay, choose destination. We click file. Then it is body for FASTA. Okay, FASTA. Then click file. We click download folder. So this is where it is available in the download folder. Right click on this and you click show in folder. So download this sequence. So we just come here, right click and we select desktop. We select desktop. So desktop. So this is the sequence. It is the name of the sequence. Then by right clicking it and reading. This is option. So we like the option because the same file we said download both. Now you see how it is here. That is when you find it. This is the file. So it has started name will be changing because the COX1 sequence already we have labeled. Do not try COX1, simply write name of it. Okay, lack of time. So today we shall done after